slack. We are ready for our next speaker, Tim Peterson. Where are you, Tim? Thanks, Tim. Yep. Let's give a welcome to Tim.
So hi everyone, thanks for being here, and I'd just like to tell you about the chemistry that unites infectious disease and aging. So hopefully I'll interest you in how we should care more about infectious disease. Okay, so just some background, and the background's a little bit, it'll seem a little bit of a non sequitur, but hopefully I'll make you appreciate kind of how we got to where we got, and I'll go through kind of the bulk of the presentation. These types of drugs that we call CADs, cationic amphiphilic drugs, their relationship with sphingolipids, which is kind of the central biology of this talk, how those connect to pathogens, and then lastly how that connects up with aging, and then of course the big picture of why we should care about infectious disease. Okay, so the little bit of a detour to start with. So I'm very interested in drug mechanisms. So I think with small molecule drugs, there's always this kind of lock and key mechanism we think happens, and of course biology is not that simple. So there's a lot of mislabeling of drug mechanisms, and I'd like to tell you a story a little bit briefly about antidepressants and their relationship with serotonin, the very famous neurotransmitter. Okay, so I became interested in SSRIs, selective serotonin reuptake inhibitors. These are widely used drugs, and became interested in these drugs because they of course help a lot of people, but mechanistically what happens doesn't really make sense with these drugs. So they're very potent inhibitors of the reuptake process in the presynapse, so serotonin stays more in the synaptic cleft, and presumably, that's really what's providing for their therapeutic mechanism. But they don't work in half the people. So many people will just keep taking different antidepressants. Psychiatrists will administer another one, and there's no real kind of, it's just really a trial and error process. So that if the molecular mechanism is so precise, we think we understand so precisely, why are we just kind of doing a Russian roulette with the therapies? That doesn't make any sense. And also the drugs take many weeks to work. So again, if the mechanism is immediate, how is this drug taking many weeks to work? Several, 2-3 weeks if it's going to work at all. So those things don't line up with the kind of monoamine basis of depression. Okay, so we've long basically hypothesized that that essentially there's kind of a disconnect between serotonin and depression. This really pivotal study just recently came out that there's never a nail in the coffin, but essentially this was pretty much the most extensive piece of work that emerged to analyze all the studies on the relationship between serotonin and depression. Essentially, the take-home message is kind of highlighted in the center here is essentially that there's really no relationship between serotonin and depression. So all these drugs are, they do work, they can work. Definitely not trying to discredit the field, but that there's really no evidence between serotonin and depression. Okay, so like I said, a bit long mission drug mechanism of action. So we have a variety of approaches, mainly genetic approaches. So there's a little bit of an irony here. I mean, I'll focus on chemistry, but ultimately to get to the chemistry, we mainly deploy genetics. So we've named kind of our kind of suite of approaches at my company, biochemistry. Mechanism of action technologies are remote, kind of drawing a moat around what you find most interesting. Okay, so kind of as I alluded to with moat, what we're really talking about is that small molecules, in many cases, don't claim what people think they target. So we do all these biochemical assays, we do these binding studies. Many of these studies, a shocking degree of them, really are leading us astray and telling us things that we kind of cherry-picking findings. So really our approach starts with genome-wide, unbiased CRISPR screens. We've done RNAi before, been in the space for a long time, use haploid-based approaches and others. But essentially it has to be an unbiased, has to be a genetic-based approach. We use self-fitness as the phenotype, so again, a kind of a very agnostic phenotype. And really the work, I owe a lot to several people that kind of got us in this field. Tijn Bromelkamp at the Netherlands Cancer Institute, Jonathan Weissman, Jason Seltzer more recently has a really nice paper on cancer drug mechanisms that I'm showing here. So how does this work? So we started with the osteoporosis drug bisphosphonates. So these are one of the most widely used drugs for osteoporosis. So this is pretty much a dead field to most people. Most people completely do not care about these drugs. They work phenomenally in a lot of people. So the question is, why would we ever do an experiment with these drugs? They target this enzyme in the cholesterol pathway, mevalonate pathway called farnesyl diphosphate synthase. There's really nothing interesting happening here. So we applied our genome-wide CRISPR screens to this approach, and when you do these screens, you mutagenize the genome, you treat them with drug, you allow them to grow, and basically get this kind of Darwin in a bottle, evolution in a bottle, where you get different pools depending on of cells, single gene mutations depending on whether you've treated or not treated the cells. You get a heat map, or sorry, a volcano plot of all the data. You get the sensitizing hits, so the cells that deplete in the presence of the drug, and then the resistance hits, the ones that behave as if the drug is not there. So each one of these dots is a gene in the genome. So what's really exciting about these approaches is essentially the genetics, in many cases, will essentially put a bullseye right exactly on where the drug is targeting. So when we did this, we were excited to see, so there actually had never been a study that had, in an unbiased manner, determined that farnesyl diphosphate synthase was the target, but essentially the pattern of hits flipped from resistance to sensitizing back to resistance, and it essentially can draw a bullseye right around this particular enzyme in the cholesterol pathway that pretty much unequivocally, really the top hits were all these genes that kind of unequivocally associated this gene with the drug mechanism. So again, genetic approach, but in pretty much all cases we've looked, and I'll go through this with antidepressants and single lipids in a second, really establishing, essentially making a relationship between the genetic data and a physical interaction between the drug and the target. And of course, if you're doing research, you want to find interesting things, and so farnesyl diphosphate synthase essentially is competitively inhibited by these drugs, so it takes a pyrophosphate, a farnesylated pyrophosphate, and so the bisphosphonate drug blocks that interaction. We also found a couple other genes that essentially also take into account organic phosphates. So SLC37A3 actually is a glucose 6-phosphate antitransporter. So mechanistically, essentially, the drug is plugging this transporter, and this is really just a completely different mechanism of action that we identified through this. And so that led to several papers. We've gone in vivo, and all this checks out, despite the fact that we did these studies in a leukemia cell line, totally irrelevant context, but ultimately established that the new biology we discovered was relevant to osteoporosis. Okay, so applying this approach to antidepressants as well as other kind of clinically relevant drugs. So we started with antidepressants. We did basically many CRISPR screens with these drugs, and essentially what emerged as a result of this is kind of this data is just a meta-analysis of the top hits, top sensitizing hits, and here the pattern was just very clear. I mean, there was really just almost no other signal. There's really everything related to sphingolipids. So serine pulmonyltransferase is the initiating enzyme in sphingolipid biosynthesis. Just far and away, massively significant, statistically significant hit, and several other factors supporting the sphingolipid metabolism were also top hits. The results were incredibly selective. So we've, of course, many people have done many different CRISPR screens on a variety of molecules. We've done dozens ourselves, and so really the pattern of these kind of top hits, is shown in this heat map here, are highly specific to what we're calling CAD drugs, these cationic amphiphiles, which include the antidepressants. So just kind of a little bit, now I've mentioned this a few times now, so just what are these CAD drugs? So like I said, antidepressants are part of this type of drugs. They also are known to some people as lysosmotropics. And so really, it's actually pretty simple. So they have, in terms of the chemistry, they really have two defining properties. So their C log P, essentially how hydrophobic they are. They're kind of octanol. Water partitioning is greater than 2. So they tend to be kind of greasy. The antidepressants, they go through the blood-brain barrier very well. So that hydrophobicity is important for that. And so a lot of them tend to be neuro or psychiatric acting drugs. They also have a high pKa, so that means they're more basic. The lysosmotropic part makes it sound like lysosome, and indeed these drugs get protonated when they get into acidic organelles, get trapped there, disrupts single lipid metabolism, and that's also part of the mechanism. Here's just kind of the structures of what some of these drugs look like. Maybe hard to make a whole lot of sense out of this, but essentially, again, there's always this kind of highly hydrophobic part, and always an amine that is protonated. So high C log P and high pKa. So actually quite basic, and so all these drugs, sertraline, which is Zoloft, fluoxetine, which is Prozac, all these kind of very famous antidepressants are in this category. Okay, so just the summary for part 1 here. So essentially the take home from when we did these CAD drugs, including antidepressants, we essentially found a variety of some of the most important single lipid related genes. And in terms of, often we do these screens, like I mentioned with bisphosphonates, usually you end up finding, even though they're genetic screens, you end up finding the chemistry in the cells that kind of lines up most strongly with the genes that you found. And so in this case, in terms of the CAD drugs, CAD drugs are amphiphils. Sphingolipids are also amphiphils. It's really targeting essentially there's a competitive inhibition happening here, very similar to what we saw with the bisphosphonates and the osteoporosis. So, and then I think in terms of the drug mechanism of action, just a broader point that we really are really bullish on the idea that genetics, in particular, can help elucidate a lot of drug mechanisms, and we've done this in a variety of other contexts besides what I'm telling you here today. Okay, so how does this all relate to pathogens and infectious disease? Okay, so I was very fortunate to work alongside Tijn Brummelkamp and John Corrett when I was back at MIT back in the day, and they really were pioneers in the genome-wide screening space in mammalian cells. They started with infectious disease, largely because those stories were quite simple. You'd find the receptor, you do a genome-wide screen, you'd add a virus or a bacteria toxin that would be toxic to the cells. You do the screen to knock out host factors one by one, and you'd pretty much immediately find a receptor that would be required for the cytotoxic effect. And so many of these Nature papers are all theirs. And so and I followed their work over the years. And I think really I just looked through when COVID hit, just looked back through their papers, and as well as by others in the field, and said, well, kind of what are the common themes if we look at all these pathogen screens? What are the common human genes that emerge from this work? And I was really shocked because essentially it's the exact same list of genes that we saw when we did our screens with the CAD drugs. So it's SPT, the COG factors, VPS factors, other kind of core sphingolipid machinery were really the top hits. And here's just the kind of heat map of the result. Essentially 80% overlap of the kind of meta-analysis of the pathogen screens and the CAD screens that we had done. So again, highly specific. These drugs seem to be really acting on the same factors that others have seen when they do screens with pathogens. Okay, so just brief summary of what we've connecting it all up here. So it's really, again, sphingolipid genes seem to be really important for not only the CADs, but how pathogens do what they do to our cells. And then, when COVID hit, like I said, I was like, well, somebody should test this in in the case of COVID. And I was very fortunate that I have a colleague in psychiatry, Eric Lenz, who's a very ambitious professor, and he actually tested fluoxamine in COVID. And to my knowledge, this is really one of the only repurposed drugs that have kind of shown promise. He and others have validated this as a strategy for COVID. And they have a JAMA paper from 2020 on that. So it's pretty cool to be part of that story. Okay, so all right, so how does this then all relate to aging? Okay, so we did, like I said, I'm very bullish on genetic approaches. So not only CRISPR screens, but we also did transcriptomics data. So we're interested in what the transcriptional profile looked like with both CADs and pathogens. And when you do that analysis, meta-analysis on a variety of pathogens, a variety of CAD drugs, again, basically identical profiles, huge number of senescence factors, huge number of sphingolipid-related genes. So really, it really suggests a very strong overlap of sphingolipid biology and senescence biology that's connecting this all up. And you can find all your friends here on these diagrams. Okay, and then so lastly, in terms of the kind of the new data, we're of course, we think we've kind of unlocked a broad category of senolytics. So we've done a variety of assays, these senescence assays in vitro, and pretty much every time it's worked, we have dozens of examples of these, very potent and highly selective effects in senescent cells with these CAD drugs. Really very little effect on proliferating cells over orders of magnitudes of concentration. So very potent and selective effects. And then a variety of pretty much no non-CAD drug we do ever works, and that's probably consistent with a lot of your experiences. So to summarize here, part 3. So it's really the chemical structure that predicts the senolytic activity. And we think this is a pretty important chemical structure, pretty important paradigm that we've established here. And we did those senescence assays not actually in the context of pathogens. We did them with DNA damage, like a lot of you all, I'm sure, do. So we really think these CAD drugs would work in a variety of senescence induction paradigms, and so we're actively exploring this. So just in the big picture, just to kind of finish up here, so I think we need to not forget that how important infectious disease is for aging. So here's some of our own data, just to that really was striking to me. So if you're in the aging field, you looked at GWAS studies, you've probably seen ApoE emerge, from a lot of those studies. So we did a meta-analysis of the UK Biobank, taking all the top aging-related diseases, try to again figure out what the common genes were. It's not only ApoE, it's not only insulin receptors, so those things that we already know about, but really a variety of receptors and other factors that are important in how pathogens do what they do. The herpes receptor, herpes affects over 2/3 of the world's population. So receptors to that are actually, in terms of statistical significance, are more important than many of the genes we already care about. So, huge plug for the factors that are controlling our response to infectious disease. And then lastly, Preston put forth this relationship between life expectancy and GDP, and of course that's increasing over time, but I think we need to not forget, how did we get from only living 30 years to now 70, 80 years? It hasn't been through these fancy medicines that we've come up with. It's largely been very basic things like hygiene and antibiotics and things that were, public health initiatives that helped us thwart infectious disease that have led to the biggest increase in life expectancy. And so we're seeing what we're doing right now is kind of the 21st century version of that. And just acknowledge some people that did the work. Thanks for your attention, and any questions, happy to talk.
Thank you so much, Tim. Do we have questions? We have a question down there.
Hi, nice talk. I just have a little criticism. I would say I wouldn't be so dismissive of other people's work, because you do your lentiviral screen in cancer cells. This is K562. Mm-hmm. These are not neurons. You can't really say what you said, serotonin is not working. you have to validate it in primary cells, then in neurons, you know.
Yep.
So this
Yeah,
kind
so-
of stuff. And
Yep.
K562 is the cancer cell line. Cancer cells are kind of crazy. So,
Yep.
and another thing is when you administer a drug, it's in the organism, right? So not just one type of cells. There may be contribution from the liver, you know, organs that do xenobiotic metabolism. So I would just put this a little grain of criticism into your study and wouldn't be so dismissive and saying this is not how it works.
Is there a
I mean,
question?
let's appreciate other people's work. And yeah, the question is whether you validate it in primary cells and in the target cells.
Yeah, so Eric Goldman's at University of Essen has done a lot of that work, so you can look up his work. His work preceded this work, and so several people, yeah, of course, no one's trying to, the definitive studies, you know, will never be done. I mean, we'll do our best to kind of make conclusions, but yeah, we're certainly not trying to rule out the serotonin hypothesis. We're just trying to promote the importance of sphingolipids in the mechanism. So, thanks.
All right, thank you so much. I think we have to move on, but thank you very much, Tim.